Hello, today I'm going to talk about the construction and the manufacturing of soil as part of a terraforming project. First of all, the biological terraformers, these are the people that are actually going to be using life to terraform, particularly Mars, will have to wait while the atmosphere is built, radiation protection improved and surface temperature corrected in case alien organisms appear out of the regolith. This includes viruses and even short-string viroid-like structures under the definition of life. The period of physical terraforming could easily be 500 years, but would allow the biological terraformers to plan an experiment in the run-up to introducing the first life from Earth. In fact, the physical terraforming process may not be over, and it may only be part way through, so the atmosphere may be very thin, temperatures may still be very low, but just high enough for some life from Earth to exist. So why is soil important to terraforming? The answer is that Mars regolith has very poor water retention capabilities, so it would be dry even after the heaviest of rainstorms. The rain would just rush through the irregular rock and sand particles. Turning regolith into soil is really critical to biological terraforming. However, if it's not done, then the whole terraforming project would be for nothing. But add organic material and life like bacteria to worms, and then it would support the growth of plants, and plants means we can eat and animals can eat, and humans could potentially exist on the surface. The first organisms to introduce to the regolith on Mars are probably going to be Extremophiles. The low temperature limit for bacteria, archaea and unicellular eukaryotes is about minus 20 degrees Celsius, that's about minus 4 Fahrenheit. Lichens are able to grow down to about minus 10 Celsius, that's about 14 Fahrenheit. Higher plants and invertebrates can survive down to minus 70 Celsius, minus 94 Fahrenheit, but for the completion of their life cycles, multicellular organisms have to have a limit of about minus 2 Celsius, 28 Fahrenheit. Mars temperatures are on average about minus 63 Celsius, that's minus 81 Fahrenheit, which is far too low for these organisms to survive, grow and multiply. Biological terraforming would be performed in two ways, The planet could be biologically terraformed without oxygen as an anaerobic environment, or with oxygen as an aerobic environment. On Mars, it would probably be easier to warm the surface and liberate carbon dioxide from the regolith and from the polar ice caps, and so create an anaerobic, airless carbon dioxide atmosphere. But an anaerobic carbon dioxide atmosphere would make it very difficult for many of the life forms from Earth to survive. So this would probably create a low biomass microbial ecosystem, whereas aerobic terraforming with oxygen would produce an oxygen-rich high biomass eukaryotic, that means multicellular for the most part, ecosystem. Whoever pays for the terraforming will probably want a shirt-sleeved aerobic environment as the end product, because that means that humans could survive. Otherwise, they will be reluctant to release the huge sums of money required. Whether the final product is carbon dioxide-rich anaerobic or oxygen-rich aerobic, planet Earth will have to be searched for the extremophile organisms which can survive the harsh conditions likely to exist on a partly terraformed planet. The extremophile organisms will need to be matched with the environmental conditions in their new home. This will include temperature, pressure, acidity or alkalinity, water, oxygen availability, radiation, salinity, as well as the amount of organics and oxides in the regolith. The wild rather than cultured forms would have the advantage of genetic diversity in their genetic material and may be adaptable enough to be able to be used in a variety of different environments. However, some of them might need to be genetically modified to cope with conditions that are very different from those on Earth. These organisms may also need modification so as to grow under extremes of temperature, air pressure, acidity or alkalinity, water availability, high radiation levels, low oxygen availability, high salinity, low organic soil content, or low nutrient availability. They may also need enhancements to their abilities to perform certain tasks, such as releasing higher levels of oxygen, nitrogen, methane, ammonia, or other greenhouse gases from rocks, or altering the albedo, the reflectiveness of the surface, or increasing the organic content of the soil, or tolerating high levels of toxic chemicals. Now I want to talk about the aerobic biological terraforming of Mars and start with the coldest places on Earth. These are in the land masses nearest the poles. From the South Pole, great distances need to be traversed over icy terrain before reaching the ice-free fringes of the Antarctic continent. At the North Pole, the Arctic Ocean has to be crossed before land is reached. The Arctic Ocean in the North Pole keeps these areas of land warmer than the Antarctic by transporting heat from warmer ocean areas. These very cold places might be useful sources of life forms which could be seeded into the warmest places on a partly terraformed cold planet. On Mars, the equator will probably start to warm up before the poles, and that lower altitude places might have greater water availability, air pressure and temperatures. So that suggests that we actually start by trying to build soil in deep craters. There is a toxicity problem on Mars due to the presence of oxidizing percolates and toxic levels of chromium, nickel and zinc in the regolith. This may not be everywhere, but may kill life forms in some regions. There may be a number of potential ways to deal with the percolates. They may have been mined as a source of oxygen or rocket oxidant, but the remainder is readily soluble in water, so it could be washed out of the regolith by extended rainfall. The Antarctic dry valleys, located at 78 degrees south, are about 1,332 kilometres, that's 828 miles from the South Pole, and are the coldest places with life on Earth, which makes them the most likely sources for the first life to be added to Mars. The winter temperatures in the Antarctic dry valleys can drop to nearly minus 65 Celsius, that's minus 85 Fahrenheit. The maximum summer temperatures in these dry valleys is about 10 degrees Celsius, but the average temperature is about minus 20 Celsius, minus 4 Fahrenheit. These regions are also very dry. In fact, more water sublimates from the surface than falls as snow. The soil is very saline and the organic content of the soil is very low. The species which have evolved to live in Antarctica have developed under very cold, dry and nutrient-poor environmental conditions, which are likely to change in the future as global warming is increasing temperatures faster at the poles than at the equator. Some species may survive, whilst others may not. The best way for organisms to spread across the Antarctic dry valleys is to have small seeds, spores or vegetative fragments which are easily lifted into the air by winds and storm. They then return to the ground attached to dust particles or snowflakes. To survive, most species protect themselves in Antarctica by either growing under the surface of the ground or inside rocks. This allows them to maintain a slightly higher temperature than the outside environments and to retain access to liquid water, which is in very poor supply in the Antarctic dry valleys, arid conditions. The first Martian colonists are likely to be microbes such as bacteria, fungi, yeasts, algae and cyanobacteria, which start to bind the mineral grains together and begin to change mobile sands and gravels into true soils. Some bacteria and fungi are able to use dust for transport in the Atacama Desert, which is also very dry. The early colonist species will start to retain water in the soil and to change the humidity of the local environment. This and the gradual accumulation of reservoirs of seeds, spores and organic matter will further aid soil development. Most parts of the Antarctic dry valleys are nutrient poor, but there are places such as penguin colonies where nutrients are available. The same might be true for Mars, as nitrate rocks rather than penguins are believed to exist in some places and to be good sources of nitrogen. Those species which live inside the air spaces in rocks are called cryptoendolithic and have adapted to survive the most severe extremes of dryness, radiation and temperature on Earth. The layer of rock, or on Mars laid or sprayed plastic, could be used between the organism and the outside, protects them from low temperatures, radiation and fierce winds. Some rocks, for example, quartz and sandstone, allow light through their surface layers so that photosynthesis can take place. The dominant organisms in these locations are lichens and cyanobacteria, which do photosynthesize, but yeasts, fungi and bacteria can also occur. Some species live under rocks, which are called hypolithic, including bacteria, cyanobacteria, algae, lichens, mites, nematodes and protozoa, which does provide some degree of protection, but not as much as the endolithic inside rock lifestyle. Another group of species live in cracks in rocks, chasmo-endolithic, where lichens and green algae grow near the surface and cyanobacteria deeper inside. Organisms which are able to hide in or under rocks or in the soil have no difficulty surviving conditions which would kill any normal living things on the surface. Together, these species are likely to be the first ideal choices for terraforming Mars, if they still exist because of our rising temperatures. In the Antarctic, lichens and algae begin to form stabilising crusts or rafts on the soil, which grow during the summer but can be blown away in the winter. The wind-blown pieces act as sources of seed, spores and vegetative material to start new rafts in other places in the Antarctic dry valleys. If these rafts are able to survive winter, they can be further colonised by more lichens and mosses. However, as the water content rises, so does their ability to trap and retain nutrients. Organisms are able to break down rocks by physical disruption and chemical acid attack. Further colonisation of the surviving rafts begins to increase the diversity so that more lichens, mosses and liverworts appear. These can be accompanied by small grasses and low-growing herbs. The expanding rafts can then be colonised by grazing flagellated protozoa, nematodes, rotifers, springtails, mites and tardigrades. These use a number of different strategies in low temperatures and poor water availabilities. Nematodes and tardigrades are able to enter a dormant state in extreme dry and cold temperatures. So, for example, tardigrades have actually been placed on the outside of the International Space Station and have survived. Arthropods do not have such a resistant state, and freezing is lethal to them, but mites and springtails produce intercellular antifreeze proteins that protect them. Besides seeds and spores, fragments of plant material such as leaves or pieces of stem can also serve the same function of spreading species across the Antarctic dry valleys. Antarctic life forms have several key talents which should help future Mars terraformers. Firstly, they can tolerate extended low light periods, as an Antarctic winter lasts for at least 6 months. Secondly, many can be frozen and reanimated with no apparent ill effects. That means that they could be frozen in the Antarctic, transported to the launch pad and remain frozen on their long 6 to 8 month trip to Mars. Those species which could not be frozen, like mites and springtails, would have to be transported in living, non-frozen form and kept alive on their long journeys. So, making soil will be the most lengthy and difficult phase of biological terraforming, but terraforming cannot be done without this step. Those species and others from Antarctica are the most suitable for early terraforming of a cold planet like Mars, but they are also the most vulnerable to extinction as our climate warms. Unfortunately, there are no alternatives, as nowhere else is as cold on Earth as the Antarctic. Therefore, they will need to be kept alive in living form in cold storage microorganism zoos, or frozen in frozen arks, or have their DNA sequenced in the hope that they can be reconstituted when our descendants are ready to start adding life to Mars or finding ways to decrease Earth's temperatures. But it's tough. We can't terraform Mars unless we have the organisms to make soil. That's enough for now. Thanks for watching. Bye.